Huh. Are you recording? Looks like it. Folks at home, somebody give me a heads up. Can you hear me?
Yes.
Okay, great. Richard, I see that you are on from home. I have your lab report already, and I will email it to you. How's that for a fast turnaround? Okay. Let's go ahead and do this. Let's see how we got. Let's see. Okay, let's get started. I would like to give you your exams and your labs back. If you have not gotten one, please come and see me. I don't want to be toting these around all semester. You can always email me your lab report, and Diana Wynn and Richard Trung did that yesterday, and I've already graded them, and they will get them back by email today. So that goes faster. And if you give it to me on paper, it will probably take 2 weeks to get it back, unless you make an effort to come and check with me at in lecture personally, which most of you are not doing. So let's begin by reviewing a little bit about parasites in general, and then moving on to herbivores as kind of very specialized parasites. This will be the last material that will appear on the exam that's coming up on Monday. And so today, we're going to have some new material, yes, but part of this presentation is just to familiarize you with some things that you probably are very unfamiliar with. And not all of what we talk about today is going to be material that I'm going to ask you about on the exam. So I'll point that out as we get to it. But we finished up last time talking about 4 issues that directly bear on the trade-off between virulence and transmission. That's vertical versus horizontal transmission, vector-borne diseases, co-infection, and serial passage. Okay? And horizontal transmission facilitates virulence compared to vertical transmission, because in vertical transmission, a parasite has to allow its host to live long enough to reproduce, so it cannot afford to be too virulent. Vector-borne diseases tend to be more virulent than non-vector-borne diseases, which is because the vector acts as a third party to greatly reduce the transmission barrier. The vector can go in search of a new host rather than the infected host struggling up out of their sickbed and trying to go infect someone. Co-infection with more than one parasite or more than one genetic strain of parasites fosters competition within the host and does tend to make the host sicker, because the parasite strain that has the higher replication rate is also stealing resources at a higher rate. And if 2 of them are vying for that, that exacerbates then the virulence that the infected host experiences. And finally, serial passage, which is something that we do in medicine when we're trying to culture parasites. We either take infected fluid from a cell culture and put it in a new cell culture, or sometimes we'll take infected fluids from, say, a mouse and inject them directly into another mouse. Well, that eliminates the transmission barrier altogether. And so frequently laboratory strains of parasites are more virulent than naturally occurring strains, which is something for those of you concerned about biological warfare to certainly pay attention to. So I'm just going to quickly pass through these 4 slides and get to the last thing we're going to talk about when we talk about parasites in general, which is complex life cycles. Now, remember that we have this transmission barrier. You need to find a host. Well, what if your host is seasonal? Or what if the conditions for transmission are seasonal? So let's say that you are infecting a host that is poikilothermic and winter is coming. What do? Well, all right, this is a problem because winter will inhibit the activity of your host. Your host is probably going to go dormant or something, and you will not be able to find another uninfected host to infect. So that's a problem. It's a typical problem that parasites face. A lot of times there are potential major interruptions in their life cycle, and parasites can easily die out unless they can find a way to get transmission more or less continuously. And sometimes that is just not possible with a single host. So in many cases, parasites will have multiple alternative hosts. And that allows the parasites to persist in nature. It reduces their extinction risk. And so the ability to infect alternate hosts is really important for many parasites. It's less so for most of the communicable diseases that we experience, like mumps and measles, but it's really important for many other kinds of parasites, like flukes and worms and things like that. So we have a terminology for these hosts. The primary host is always the host in which sexual reproduction occurs. And that's just by definition. That is how we agree on how to talk about these alternate hosts. Okay, the secondary host then is a host in which some stage of development may occur. There may be asexual reproduction. And so you may actually get a huge increase in the number of parasites through asexual reproduction. But you should view this simply as a way for the parasite to find its primary host so it can sexually reproduce again. There is also, in at least some cases, something we call a transport host, in which there's no development and really no effect on the host per se, but it just serves as a way for the parasite to get between the primary and secondary host. So a good example of a parasite that has a primary and a secondary host, but not a transport host, would be flukes. There are many, many, many of these parasites that live in the digestive system or the liver ducts. And sheep flukes are a really classic case. You have these wormy things that live in the bile ducts of sheep, and they pass their eggs out through the digestive system, and they need to get into another sheep. Well, they get into another sheep not by directly infecting the sheep, because sheep do not eat the feces of other sheep. So that's not a possible means of transmission. What does happen, though, is when those feces break down, snails will consume some of the waste products and they will become infected. So they are the alternate host. And snails are mobile, so they can move through pastures. And when sheep ingest accidentally a snail while browsing, they then become infected and complete the life cycle. So this alternation of hosts is actually a really common thing in nature. There are not that many cases where we have transport hosts, and I'm really sort of not going to talk about them much. But humans have possibly the single most elegant demonstration of this primary and secondary host system, which is malaria. Okay, so malaria is probably the disease that has killed more people throughout human history than any other. It's been with us since before there were actually humans. We have some circumstantial evidence that Australopithecines were afflicted with malaria, and malaria is very common in the tropics where we originally evolved. It is both a bird parasite and a mammal parasite. There are different strains. We believe that malaria strains that infect people probably originated mostly in birds, but here's the life cycle. So we'll start where they start, which is start here. Conveniently, they say start here. The mosquito bites a person, okay, and injects what we call sporozytes, which is a form of the malaria parasite. Okay, the sporozytes then spread throughout the bloodstream, and they need to find a way to multiply, because the goal of the sporozytes then is going to be to produce enough offspring so that they can get back into another mosquito and get transmitted again. So the sporozytes do this by infecting the liver. Okay, and an enlarged liver is a classic sign of a malarial infection. Okay, and then once they do that, they develop into a different stage called the merozoite. So sporozoites are what infects people, and then merozoites do a lot of damage. Okay, the merozoites then can infect liver cells and cause reproduction and kill them. They can also infect the blood cells. And that is what the classic fever and chills syndrome of malaria is all about. Merozoites infecting blood cells, sort of being released from the liver, infecting blood cells, and then using the machinery of the blood cells to make more copies of themselves. When they burst open the red blood cells, that causes inflammation. That gives you the fever. Okay, so you get this alternation of fever and chills because there are these mass synchronized breakouts from the red blood cells. Okay, so this says there is a 48-hour cycle. That's true for some types of malaria. There are actually 4 malaria strains that infect people. There's quercetin, there's tertian, and that refers to the recurrence interval of the chills and fever. Quercetin is 4 days, tertian is 3 days. But the merozoites then are asexual, and they eventually differentiate into gametocytes. So you got sporozytes, merozoites, which do the damage in people, and gametocytes. Okay. Now, gametocytes don't do anything to people, but that's the stage that mosquitoes take up in their gut when they feed on people. So if people have merozoites that have developed into these male and female gametocytes, they can get into the digestive tract and salivary glands of a mosquito and then undergo sexual reproduction. Okay, and since they are gametocytes, you might guess that they'd become gametes, and that's what they do. they become gametes inside the mosquito. Then they fuse and they undergo sexual reproduction. They form a zygote, right? And so the zygote is diploid and it produces then haploid sporozoites rather. Okay, and so that completes the life cycle. The haploid sporozoites travel to the salivary glands, they get injected. So haploid sporozoites are in the person. They're just swimming around looking for cells. They become haploid merozoites, which do the damage to the liver and the blood. And then this massive number of haploid merozoites eventually produces male and female gametocytes. The gametocytes can do nothing in people. They have to go into the mosquito. Okay, and in the mosquito, they find favorable conditions which prompts them to fuse to form zygotes and complete the life cycle. So these are diploid here. The zygote is diploid inside the mosquito. Everything else is haploid. Sporozoites are haploid, the merozoites are haploid, and the gametocytes are haploid. So what you can see then is that the sexual stage of the malaria parasite occurs in mosquitoes. The mosquito is the primary host of malaria. People are the secondary host of malaria. And so as much as you probably want to glorify your own role in malaria, you are simply the malaria parasite's means of finding another mosquito, because that's what it needs. It doesn't particularly care about you. It just wants to use you to find another mosquito. So you're a secondary host when you get malaria. Of course, that does not make it any better for the several billion people who have died from malaria over the course of human history and prehistory, but that's the way it is. Okay, and this is a great illustration of this alternation of life cycles. Okay, everything outside the mosquito is haploid. That means no sexual reproduction. So whether the mosquito is biting a human, whether it's biting a monkey or biting a bird, those are all secondary hosts. These are parasites of mosquitoes that use other organisms as secondary hosts. Okay, so this kind of alternation of life cycle is really common. Finding a secondary host is necessary for many parasites. And it is a solution to the transmission barrier in some cases. Because if a population of hosts becomes more and more resistant, and you have an alternate host to which you can be transferred, then you can wait out the resistance of your primary host and then reinfect that population later when it's more vulnerable. Okay. So questions about the parasites and their life cycles here. All right, now what I'm going to do then is finish up this whole section on exam 2. Whoops, let's click to exit. Come on, click to exit. There you go. Okay. By talking about herbivores as special cases of parasites, and I see that it didn't, doggone it. I updated this herbivore presentation, but it didn't capture it. Oh well, we'll just use the old one. So let's go back and share this screen now. And talk about herbivores as specialized parasites. Okay. So 4 basic things that we'll talk about today, and some of them you'll be responsible for, some not so much. So first, the issue of plants as food and animals as consumers of that food. This is a difficult thing for animals. Second, we'll look at the diversity of plant defenses. And we'll lay out a couple of general principles which I will want you to remember. And then we'll just take a tour of many, many kinds of plants and give you examples of these defenses. That part I'm not going to ask you about because there's too much chemistry in it, and we're not going to worry about that for the exam. So the principles that underlie what characteristics plant defenses have, that I want you to know. I don't care if you know the detailed structures of any of the things we're going to look at. So then we'll ask, based on the ecology of plants, which kinds of defenses in general might they use? And we'll look at plant herbivore evolution and the red queen to finish. This will be another demonstration of the red queen in action, kind of like the one with wheat rust and wheat, except the organisms involved are prettier. So first, plants as food, animals as consumer. Plants are the primary food of actually most animals. Most animals don't eat other animals. Most animals eat plants. Okay, and plants don't have the same set of options for resisting their parasites. And I call them parasites because most animals don't actually kill the plants they eat. They steal energy, they eat tissues, but they don't actually kill those plants in most cases. So they're kind of function as parasites of plants. But plants don't have immune responses in the same way that animals do. They do have something, and they can react to herbivores by changing their chemistry, but it's not the same level of sophistication as animals do. The other thing is that they're immobile and they can't get away. Once a parasite finds a plant, it's at the mercy of that parasite. So plants rely on what we call passive resistance. They rely on physical defenses or chemical defenses against their parasites. They don't have the option of this really complicated immune system that animals do, but they can produce very effective countermeasures when they don't want to be eaten by a parasite. Now, some of these defenses are quite costly, as you will see. All you got to do is count up the number of chemical bonds that we're going to be talking about, and you'll see that some of them are going to cost a lot. So a lot of them are not made until the plant starts experiencing damage. So the damage to the plant is often a sign that activates the synthesis of these defenses. And that means that they're plastic. They could be acclimative, in which case you can upregulate something and then downregulate it again once your herbivore goes away, or they could be developmentally plastic, in which case you activate them early and then you keep them because that's a signal that this environment is a dangerous environment that requires this defense. So some of them can be induced and then downregulated again. That's acclimation. Others, once induced, are permanent, and that's developmental plasticity. So here is why plants are not good food for animals. Plants are mostly carbohydrate, but animals are mostly protein. So that means that the most crucial thing for herbivores is not the energy that they find in plants, it's the nitrogen. There's plenty of energy in plants, they're all carbohydrate. But what animals need is nitrogen to power the construction of their tissues. So that's a fundamentally different view of food When animals eat other animals, they're eating things that have the same basic composition as they do. That's not true for plants. The second problem with eating plants is that plants don't like to be eaten, and every single plant, without exception, has some kind of mechanical or chemical defense against their herbivores. That means that animals have to invest in biochemical systems to detoxify those plant defenses, or their options for eating plants are limited to the ones that they can handle. And they'll have to just avoid many kinds of plants because they cannot afford the detoxification. So we distinguish specialist herbivores. They have detailed adaptations to neutralize the defenses of a specific type of plant and extract the nutrients they need from a specific mixture of nutrients. But that level of adaptation to one kind of plant means that they will be very ineffective if they try to eat other kinds of plants. So they're very limited in the range of things they can eat. They're also generalist herbivores, and they eat a mixture of plant species, and what they're trying to do is avoid overdosing on any specific defense. So they'll eat a little bit of something that might be toxic, but they don't eat a lot of it to avoid overwhelming their detoxification systems. They also will balance out the nutrient content of the different plants that they eat. So they may eat something that's relatively high in nitrogen, but then they may go and seek out something that's higher in carbohydrate to get the right mixture of nitrogen and carbohydrate. So these are very different strategies among the herbivores. Now, there's also an issue of relative size and how that affects whether you are a generalist or a specialist. So plants vary in size. They can be tiny, they can be big. Herbivores vary in size. They can be tiny or they can be big. Yes. Okay, so Dwayne is asking whether grass has chemical defenses. Yes, absolutely. The chemical defense in most grass, and the reason that you itch when you're around grasses, is called coumarin. Okay, and there are many kinds of coumarin, but the smell of freshly mown hay, if you go out in the fall and go into a farm field, there's a very distinct smell. That's coumarin. If you go on a hay ride, you're basically sitting in a bath of coumarin. And so if you are sensitive, you're going to itch. So yes, as I said, all plants, every plant has a chemical defense of some kind. Okay, but the relative size of an herbivore compared to the plant that it eats has a lot of consequences for whether you're a specialist or a generalist. If herbivores are small relative to their host plants, then they can just spend most of their life on that plant. Well, duh, you could be a specialist then, because you don't ever have to eat anything else. And many small herbivores are specialists. Some small herbivores are generalists also for reasons of ecology that have to do with like predators. But being a specialist is a good option for a small herbivore. And so that's like our little grasshopper here. This is actually a nymphal Schistocerca americana, which you can find in generally about April or May, oftentimes in the wet vegetation around the roadsides in New Orleans East. Here's a large herbivore relative to the plants it's eating. So they got this horse that's like 1,000 kilograms, and here's a grass blade that's maybe 3 grams. All right. If herbivores are large relative to the plants that they eat, they cannot avoid eating multiple plants. They're almost all generalists. So large herbivores like mammals, they're almost all generalists. Small herbivores like insects are mostly specialists, although there are some generalists in there too. But this relative size thing is a big structuring feature about this host-parasite relationship. Okay, so let's take a tour then of the kinds of defenses you can find in plants. So mechanical defenses simply reduce access to plant tissues, or they may make it more difficult to digest. Here's some wicked-looking spines, and notice that they're curved. And what these spines are doing is latching onto the exoskeleton of this big old herbivore here. This is a caterpillar, and basically nailing it down as if it was a Velcro strip. And here's your poor caterpillar that's more or less glued to this stem. It cannot get away. And so it's going to die. So hairs and spines are really effective. If you've ever seen an acacia tree from Africa, you'll have seen that they have spines that are about 3 or 4 inches long. Well, that's for the big herbivores. That's for the giraffes, to try to prevent too much browsing from those big herbivores. So other kinds of plants have other kinds of mechanical defenses. Silica in grass, it just basically acts as an abrasive and wears down the jaws of herbivores. And basically, if it wears them down too much, they won't be able to eat the grass and they'll starve. But mechanical defenses are costly. And so you're not going to invest in chemical defenses if you're going to shed a leaf after a week. You're going to invest in mechanical defenses if you're going to keep your leaves for a long time. Otherwise, it's not worth it. It costs too much energy. And you won't make that energy back if you shed the leaf too quickly. So this is an important point. When we start talking about the chemicals, some of the chemicals cost way more than others. And they're used in long-lived tissues. Cheap chemicals can be used in pretty much anything. Expensive chemicals, you're not going to use them in disposable leaves. You're going to use them in long-lived tissues. So let's talk about the general types of chemical defense then. There are 3 basic types. There's the phenolics. These are large, really expensive, and they're long-lasting cyclic compounds. They're derived from cellulose, and they are the classic expensive defenses. And then there are 2 types of less expensive defenses. First is the nitrogenous compounds, and there's tons of them, but they tend to turn over pretty quickly because if they have nitrogen, they're tied into nitrogen metabolism. Sometimes the plant can invest in a nitrogen defense, but then it'll change its mind and say, I need that nitrogen for growth. And so it'll metabolize it and reuse it. So these defenses are rapidly turned over. And so you can use these pretty effectively in ephemeral tissues. If you only want to have a leaf for a couple of weeks, you might use these nitrogen compounds, but you would never use phenolics to defend them. So here are some of the general types. You got your alkaloids like caffeine, nicotine, and some that I'll show you. You got cyanide-related compounds. You have non-protein amino acids and some peptides. And I'll give you examples of each of them. And then finally, you have terpenoids. Terpenoids are ring compounds that have a different biosynthetic origin than phenolics. They're based on a 5-carbon molecule called isoprene. And basically, you take these 5 carbon units and you put them together like Legos, and you make bigger and bigger and bigger molecules. So some terpenoids can be very, very cheap, and some can be very expensive. Terpenoids do not have nitrogen in them as a rule. So these can be small, inexpensive nitrogen defenses. These can be small, inexpensive carbon-based defenses. Okay, I want you to remember that there are these 3 general types of defenses, that phenolics are large and expensive, that nitrogen compounds are small and cheap, and that terpenoids can vary depending on their size. That's something I want you to remember for the exam. I'm going to show you examples. I'm not going to hold you responsible for any of the specific examples we're going to run through real quick. So first, let's look at those phenolic compounds. This is what they typically look like. This is the biosynthetic pathway. And notice it's a 6-carbon ring. Okay, this particular thing is shikimic acid. But look at all the hydroxyl groups on it. This is the secret to their function. Hydroxyl groups form hydrogen bonds with other chemicals. And so if you have something like this is actually something that plants use for defense. It can bind one chemical here on this one, combine a different chemical here, combine a third one here, and it combined 4 different chemicals in a big indigestible mass. Because if each of those compounds requires a different enzyme, how are those enzymes going to get at the active sites if they're all bound up in a big clump by these phenolics? So that's how they work. This is what we call a hydrolyzable tannin. And we won't go into why, we'll just go into the way that it works. And that's that these hydroxyl groups bind to other compounds and prevent them from being digested. There's a second kind of phenolic called condensed tannins. That's what this kind of thing is. And notice that it's got even more hydroxyl groups. So this is possibly going to be able to make a even huger indigestible mass. So what these things do is bind to proteins in the digestive system of herbivores. And they prevent proteases from digesting them. So the herbivore can eat the plant, but it can't get any nitrogen. They can get plenty of energy, but they still starve to death for lack of nitrogen. Okay, so we call them digestibility reducers. They're very effective against both generalists and specialists. And because they're so expensive, they're typically found in woody plants that keep their tissues for a very long time. So a tree that sheds its leaves annually will often defend them with phenolics. A tree that keeps its leaves for multiple years will also often use phenolics, like fir trees, like pine trees. Question in class. Yes. Yes, these things are often color water dark, and they're called tannins because they were discovered in tanneries. This is what people used to make leather, and it was only later that we came to appreciate what their role in plants actually was. So these are the big expensive ones. You will not find them in like a garden herb in general. It is possible to find some of these smaller hydrolyzable tannins in more ephemeral leaf tissues, but generally these are defenses for woody plants. Here are some alkaloids that you would find in your garden plants. Okay, and I've got a whole bunch of different families of alkaloids here. I don't care that you know any of these structures. I'm not going to ask you anything about them at all. But these are the classic plant poisons. If you want to poison Socrates, give him alkaloids to drink, which is exactly what happened. Now, alkaloids are ring compounds with a nitrogen in the ring. That is the defining feature of all of these, with the exception of caffeine and also nicotine, which aren't really ring compounds. But look at how many bonds there are compared to the phenolics. Now, there are fewer bonds. So they're cheaper. They're cheaper to synthesize. And that's why non-woody plants and plants that shed their leaves frequently, they use these things. The limitation on things like alkaloids is whether there is enough nitrogen in the soil to allow for standard primary nitrogen metabolism, like protein synthesis, and have some leftover for these. So if nitrogen is abundant in the soil, you're going to use these. If nitrogen is not abundant in the soil, you may have to look at things that don't contain nitrogen, like phenolics. Okay, but when soil nutrients are abundant and photosynthesis is limiting, then you don't have a lot of energy to make bonds. Hey, it makes sense to make these small compounds that are extremely toxic. Okay, so generalists generally cannot deal with alkaloids, and they often have to avoid these plants. But specialists can actually use these alkaloids to identify the plant that they can eat, because they specialize in detoxifying the particular alkaloid in their plant. Okay, so here's some examples. Here's the Socrates alkaloid. This is coneine, and it's found in Queen Anne's lace and hemlock. Really high concentrations in hemlock, but you can actually extract it from carrot tops. It's really small quantities, but it's in there. It's just not in there in enough of a concentration to hurt you. Okay? So I don't care that you remember this. I'm just entertaining you at the moment. So here's another kind of nitrogen defense, is cyanogenic glycosides. Okay, and as the name implies, there's cyanide in here. And the cyanide will kill pretty much anything. And you may ask, well, how do plants use cyanide as a defense and not kill themselves? That's what the glycoside part is. Plants use these glycosides here to inactivate the cyanide part until their leaves are damaged. When the leaves are damaged, an enzyme is released that then cleaves the cyanide off of the rest of the molecule, and the cyanide is free to go and kill whatever's eating the plant. And then notice that this thing is basically benzene. It's benzaldehyde. And so you frequently get the smell of sweet almonds when something is cyanogenic. You're not smelling cyanide because that's extremely bitter, but you're smelling the benzaldehyde that's released when the cyanide is freed from the glycoside. So these are found in the seeds of fleshy fruits and apples and peach pits. Don't eat those because you can die. Okay. Now there is a enzyme that detoxifies cyanide, and that's called rhodanase. And notice that it's rhodanase. It's not rhodanase, as most is true with most enzymes. There are organisms that eat highly cyanogenic food, and they have huge amounts of rhodanase in their tissues. Question in class. Okay, so once the cyanide is released, can that tissue still function? No, the tissue is going to die. This is the plant realizing that it's It's going to lose this tissue permanently, and so there's no harm to the plant in releasing cyanide into something that's going to get eaten anyway. So again, entertainment. I want you to know that there is such a thing as a cyanogenic glycoside, and I want you to know how plants avoid killing themselves. That's, binding the cyanide to a glycoside. Here's another cyanide compound. This is an isothiocyanate. This is a mustard oil. This is what gives mustard its flavor. When you use Dijon mustard on your sandwich, you are basically poisoning yourself. Okay, and what you're smelling is the sulfur compounds that help the plant avoid toxicity from the cyanide. So here you see that you've got cyanide and it's bound to sulfur-containing compounds. And so when the plant is damaged, you get enzymatic release of the cyanide plus these sulfur compounds. That's why mustard smells the way it does. And these are called mustard oil glycosides because they are found in the mustard family. So, a couple of kinds of cyanide here. Now, non-protein amino acids are really interesting, and they're found generally in legumes. And here's a legume. This is Cannivalia, and it's got a big old seed pod here. And you might think those seeds might be good to eat because a lot of our seeds are edible, but in this case, that would be a mistake because these seeds contain a non-protein amino acid called cannavanine. And it takes the place of arginine in metabolism. And unfortunately, there is a difference here in these two amino acids, and cannavanine inactivates any protein in which it is incorporated. So it doesn't kill you immediately, but over a relatively short period of time, it destroys your ability to synthesize functional proteins. And that should be a warning sign to you. So I don't care that you know this. I care that you understand that these non-protein amino acids substitute for the good ones and produce non-functional proteins. That's all I care about. Okay, how about some peptides? Well, I have an example of Amanita deathcap mushrooms, and they produce these peptides here, and they basically destroy your liver. The modified presentation I wanted to show had ricin and abrin, which are actually produced by plants, since fungi are more closely related to animals than plants. I wanted to get a plant in there, but it's the same mechanism. They destroy the liver, and so you eat this stuff, you get sick, you feel better for about 2 days, and then you die. All right, now let's look at terpenoids. These are polymers of isoprene, and here's a 5-compound isoprene. Okay, and look at the shape. So you could put 2 of these together, and you can make a cyclic compound, and that's how we name terpenoids. Here's some monoterpenes that are made from 2 isoprenes. These are the aromas of plants. Myrcene, limonene. I bet you can't guess what plant limonene is found in. Pinene. These give herbs their odors. So when you use herbs in cooking, you are poisoning yourself. But not to any great degree because you're not using enough. But these things are not there to make your food taste better. They're there to kill the insects that eat these plants. So sesquiterpenes use 3 isoprene units, and here's some examples of those. Again, I don't care that you know any of this. I'm just entertaining you at this point. Here's diterpenes. Okay, that's 4 isoprene units, and oh, that's starting to look like steroids. And in fact, some of the triterpenes are steroid mimics, and they can interfere with steroid metabolism. Okay, here's something called azadirachtin. Azadirachtin is a tritropine that actually binds to your salivary receptors and inactivates them. So you can't taste anything. And in insects, that's pretty fatal because they stop eating and die. Okay, so here's triterpenes as steroids. And you can see that if you get the wrong kind of triterpene defense in there, it can really mess up your steroid metabolism. Okay, so here's the relationship of cholesterol, estradiol, cardinalides. Cardinalides are cardiac poisons. That's what's in milkweeds. Okay, and if you chomp on a milkweed, you'll cause heart palpitations because they are potent cardiac poisons. They're also found in oleanders, so don't eat oleanders either. Typically, milkweed butterflies can detoxify this stuff, but you can't, so don't eat it. Okay, here's digitoxin, and these are all examples of complicated terpenes. Here's digitalis or foxglove. This is actually used in pharmaceutical research, and it's the basis for treating cardiac arrhythmias. So not all plant defenses are harmful. They flavor our food, they provide medicine. Okay, finish up, last thing to say. The kind of chemical defense that you're going to use depends on your characteristics as a plant. If you're a large plant and you cannot get away, then you have relatively permanent tissues because you're large. So you're more likely to use things like phenolics that are very costly. If you are limited by the ability to photosynthesize, though, like if you're growing in the shade of an understory in a forest, then you may be able to make use of nitrogen in your defenses more than making big, complicated molecules that require a lot of energy. So this trade-off between resource availability in the soil and photosynthesis can shift your defenses from carbon-based to nitrogen-based. Okay, so if you're limited by photosynthesis, use the small nitrogen compounds. If you're a small plant and you're not limited by photosynthesis, maybe you can use the terpenoids, especially if nitrogen is limiting in the soil. Okay, but here's the last thing. Here's another example of the red queen in operation. This is the umbellifers, the plants that include Queen Anne's lace. And here's an example of how they've evolved over time with their butterflies. These are found in the basal plants of the umbellifery clade. And they have their own butterflies, right? And the butterflies have developed detoxification enzymes, and so they can eat the plant. But some more advanced members of that group evolved different compounds, which these butterflies could not eat. They're toxic to these butterflies. However, then you had some plants that were an open resource, and any adaptation in the butterflies that allowed them to metabolize this stuff would allow them to have a resource without any competition. And so sure enough, over evolutionary time, some of the butterfly species develop detoxification enzymes and can eat these now. There are two different elaborations of this that are found in the most advanced kinds of umbellifers, and that's linear and angular foranicoumarins. This is exactly the right shape to get into a DNA double helix and inactivate it. And only the most advanced butterflies have the enzymes that can detoxify these things. So the butterflies that feed on these plants cannot eat anything else. The butterflies that eat these plants can eat these, and they still have the ability to detoxify this. The butterflies that can detoxify these things have the ability to detoxify the whole sequence. And so we know that the earliest evolutionary representatives had the simplest defense and they had the simplest detoxification enzymes. The plant got an advantage, the evolutionary arms race. But the butterflies caught up. The plants got another advantage, and the butterflies caught up yet again. And we can expect this to continue on in ways that we probably can't predict right now. But it's a second really nice demonstration of the Red Queen in action over evolutionary time. Every time the plant gets an advantage, the butterflies counter with their own measures. The plants evolve again, the butterflies follow along. So this is a case of more stepwise rather than simultaneous evolution in the Red Queen. And that's all I've got. Do we have any questions, either in class or at home? If we don't have any questions in class or at home, then we are done. And this is the end of the material for Monday's exam. Okay, I will be available for consultation in office hours or over email or Zoom whenever you need it.